Inside every living cell, millions of microscopic machines are constantly in motion. These are proteins, executing every critical task required to keep biological systems alive. Every protein begins its life as a simple, one-dimensional chain of building blocks called amino acids. But a flat chain cannot perform work. To become functional, it must fold into a highly specific three-dimensional architecture. In biology, the shape of the protein dictates its entire function. When a protein misfolds, the biological consequences are severe. These structural failures are the direct cause of many human diseases, ranging from Alzheimer's to cystic fibrosis. Human health is governed by this shape-based biological language, a code that scientists have spent over half a century trying to reliably predict. While we can easily sequence the one-dimensional chain of a protein, predicting how it will twist into 3D is a mathematical nightmare. The number of potential shapes a single protein could theoretically take is astronomical. Finding the correct fold through random trial and error would take longer than the age of the universe. This physical time constraint makes brute force calculation impossible. To bypass the math, traditional methods rely on physical observation. Scientists use massive equipment like NMR spectrometers and X-ray crystallography to freeze and map these structures by hand. These experiments are grueling. Mapping just one protein structure can require years of laboratory labor and immense financial resources. This physical bottleneck has historically stalled molecular biology, slowing the development of new medical treatments to a crawl. In 2020, at a global competition called CASP, a system developed by DeepMind called AlphaFold demonstrated that this biological bottleneck could finally be bypassed. AlphaFold reached these results through deep learning algorithms, working without the physical microscopes or wet labs that define traditional research. The system was trained on tens of thousands of known protein structures, with decades of human experimental data flowing into its neural network. By processing this dataset, the AI learned the hidden physical rules and patterns governing how amino acids fold. This approach turned the protein folding problem into a data-driven translation task, moving the work from the lab bench to the processor. To process entirely new protein sequences, AlphaFold uses a specialized architecture called an attention mechanism. This allows the system to scan a long amino acid chain and selectively focus on distant parts simultaneously, deducing how they will physically relate in 3 dimensions. Guided by these spatial relationships, the chain collapses into its final architecture in a digital simulation. The accuracy is high enough that these digital models now match the quality of physical experimental data. Paces that once required years of grueling physical labor in the laboratory are now achieved by the system in mere minutes. AlphaFold acts as a dimensional translator, successfully bridging the gap between a simple one-dimensional sequence and its functional 3-dimensional shape. Providing researchers with instant, accurate 3D blueprints allows them to see exactly how new chemical drugs will interact with target proteins in the body. This clarity speeds up the drug discovery process, particularly for developing highly targeted cancer therapeutics. It also provides the first structural views of uncharacterized proteins linked to rare diseases, which were often ignored in the past due to the high cost of studying them. Computational data is now being converted directly into a pipeline for tangible medical treatments. Limitations still exist. Predicting massive protein complexes and membrane-bound proteins remains a challenge that requires ongoing algorithmic work. There are also ethical concerns, as making detailed biological blueprints openly available requires frameworks to prevent malicious use of the data. AlphaFold provides a high-speed starting point for researchers, allowing them to verify AI-predicted models through targeted physical lab work. This system has moved molecular biology toward a high-speed computational discipline, where human-AI synergy drives the pace of discovery.